Can people hear me? Oh, there you are.
Yep.
Okay, great. Are there any questions before we begin? All right, I'm not hearing any questions, so let's go ahead and begin. I'm going to plug in my headphones. So why isn't that going? All right, there it went. We're going to finish the lesson on chapter 7 today, and then do a little bit of review for the final. Any questions about any of that? All right, let me show you. My screen. All right, you should be seeing my screen now. So today we're actually supposed to be doing a review and catching up. We got a little bit of catching up to do. We need to finish chapter 7. We shouldn't have any trouble finishing chapter 7 today though. And then we'll do review. If I don't finish the review in the lecture, I will continue it in the lab. So there may be a short lab today. Okay, in which case I'll just be talking about reviewing for the final. There is no real lab work. You should be finishing your unknown project. The unknown notes pages will close on Friday. I think it closes at 11:59 p.m. on Friday. And then the unknown project report should be written up and turned in by 11:59 p.m. on Saturday, June 11th. The last day for requesting a test will be Friday, but understand you will only have 1 day of incubation to get that test done. So really, today is the last day for requesting a test because most of our tests require 2 days time. There is one test that requires 5 days. And so if you were to request that test today, you're not going to get the results in time. In fact, there's one student who requested the MR test a couple of days ago, and I worked it out. They're going to get the results on Saturday, June 11th, which is doable, but that's putting it pretty close. Okay. So get your last test requests in by today, and I will accept a few tests on Friday, but it will only be a few, like the catalase test and the, I'm losing the name of that one, the oxygenase test. If the test can be done in 24 hours or less, you can request it on Friday. Any questions about any of that? All right, the final is on June 15th from 8 p.m. You must get it started, and it must be finished by June 16th at 8 p.m. And let me check that just to make sure that I've got that right. I see that it starts on June 15th, but when does it end? Yeah, it ends on June 15th at 8 p.m. And hopefully that's Wednesday. Oh yeah, it says by June 8th. All right, I might have said that wrong. You can take it as early as Tuesday at 8 p.m., but it must be finished by Wednesday at 8 p.m. It's an online test. It'll follow the familiar format. There will be one test, which is going to be short answer or short essay. And then there will be more questions, which will be multiple choice. The multiple choice questions require Respondus Lockdown Browser, and it is a timed quiz. 70 minutes is the time limit on this quiz. So once you begin, you have to finish the quiz by 70 minutes, and it must be finished by 8 p.m. June 15th. Any questions about any of that? The other exam, Final A, you can start it and you'll have 24 hours to finish Final A. And that'll be the short answer and fill in the blank questions. Any question of that? All right, the correct answers will be shown about 2 days after the exam. So you can look up the questions 2 days after you take the exam. Or I should say 2 days after the exam finishes. All right, let's go back. So close that. The final is cumulative and comprehensive, but understand that about 50% of the questions will cover the chapters that you have not been quizzed on. So about 50% of the questions will come from chapter 7, which we're going to finish today, chapter 13, chapter 14, and then lab module 9. You have not been quizzed on any of these. And so there will be more questions on these, and about 50%. And you'll note that there will be questions on the final concerning the lab. So you do need to study the lab as well. There will be questions from the earlier chapters, and that'll be chapters 1 through 8, excluding chapter 7. But generally, there will only be a few questions from each of these chapters because you've already been quizzed on that material. And that's true for the earlier labs, labs 1 through 8. They will be tested on, but there will be fewer questions than on lab module 9. Any question about any of that? And there will be some questions on your unknown project. Usually what I do for that is I will give you some characteristics of a bacteria that you're trying to identify, and then you have to identify it, and I will provide a table for you. Okay, any questions about any of that? All right. Let me briefly state that I have been trying to get some of the grading done. Let me stop sharing my screen and then I'll be able to tell you what I got graded, because I won't remember unless I go to the grade book. I think I've gotten all of the quizzes graded. So quizzes 1A through 4A are now graded. And I have the earlier labs graded too, but I don't remember which. That's why I'm calling up the thing. All of the extra credit study assignments have been graded. I think all of the worksheets are graded except for worksheet for chapter 7. And then lab 0, lab 1, lab 2, lab 3, lab 4, lab 5. Well, lab 4 have all been graded. Lab 5 is slightly over 50% of them are graded. I tried to finish lab 5 last night and I just got too tired and went to bed. And I haven't gotten able to do lab 5 today, but my goal is to get lab 5 graded shortly after the lecture ends. Okay. All right, any question about any of that? Obviously lab 4 and 5 graded. We're talking about the second grade. People did really well on labs 2 and 3. Lab 4, some of the people did not do too well, and most of those people did not update the lab, meaning when I told you the answers were wrong, they didn't go in and put in corrections. I still need to grade the infectious disease research project. I hope to get that graded this weekend. And the unknown report, I don't know when I'm going to get that graded. Probably about the time you're taking the final, and that will be only if I am doing well in my grading. I need to correct quiz 5. If you remember, it was worth less than 1 point. I need to make a correction there and make this up to the full quiz points. I haven't done that yet because when I correct quiz 5, I then have to put in your drop quiz. Otherwise, you'll have too many points. And I want to do those together because students have occasionally complained that they had a higher grade and then I put in the drop quiz and the grade dropped. And that's because when you're counting quiz 5, that means you have too many points for the quizzes because one of the quizzes is going to be dropped. And I don't remember the number of points, but it's something like 300 points for quizzes. And if you count all the quizzes, it's 75 points too many. Any question about that? All right, I hope to finish the labs soon. Like I said, my goal is to get lab 5 finished tonight. And maybe even get started on lab 6, but we'll see how that goes. All right, any question about any of that? Let me shut the grade book and re-show you the screen. And let's move on to chapter 7. All right, so last time we had started talking about the chemical methods for microbial control. There are a few agents that do achieve sterility, and that's because of endospores. Most of the chemical agents do not kill endospores. Endospores are difficult to kill. Are you guys hearing me okay with the headphone?
Yes.
Okay.
Yeah.
All right, great. Disinfection refers to the destruction of vegetative pathogens. By vegetative pathogen, we're not talking about endospores. There are different factors related to effective disinfection. There's the concentration of the disinfectant. Generally, the more concentrated, the greater the disinfection. The time is a factor in effective disinfection. It has to be long enough to kill the cells. And you also have to have access to the surface of the cells to be disinfected. Like UV light, I think I mentioned it. When you're sterilizing something like this can, for example, with UV light, it would not be able to get inside the can. It would only sterilize the surface. And it wouldn't sterilize the bottom either. So UV light is very good for sterilizing surfaces, but not inside of, I don't know, objects or whatever. The pH will affect the disinfection. Generally, more acidic pH, the greater the disinfection. And the presence of organic matter can decrease the disinfection. For example, bleach is a very good disinfectant, but it is inactivated by organic matter. And I'm mentioning bleach, but understand there are many disinfectants that are inactivated by organic matter. Any question about any of that? And of course, organic matter is important because when you're dealing with a clinical specimen or a clinical situation, most of the time you're going to find organic matter with the bacteria. There are different types of disinfectants. There are phenol and phenolics, biphenyls, which are similar, biguanides, halogens, alcohols, excuse me, heavy metals, surface active agents called surfactants, and soap is actually considered that, hand soap, chemical food preservatives, antibiotics. And we're going to only talk in this lesson about antibiotics which are used as a food preservative. We're not really going to talk about other forms of antibiotics like penicillin G. There's aldehydes, there's gaseous chemosterilizers, and the gaseous agents are very effective at sterilizing. So they can kill endospores. The trouble with gaseous agents like ethylene oxide is that it takes a long-term exposure to kill bacteria, particularly endospores, but they do kill endospores. And another problem with gaseous agents is that they're highly toxic to us. So you wouldn't want to like sterilize food with a gaseous chemosterilizer. And you don't want to expose people to it either.
Dr. Volpe,
Uh-huh.
does that also kill the prions?
No. Prions are very difficult to kill. Gaseous agents and peroxygens do not kill prions. Prions are not exactly alive, so killing isn't the right word. So we'll say inactivate. They do not inactivate prions. To inactivate prions, I stated that last time, you need to heat the prion infected agent to 1,000 degrees Fahrenheit and incinerate it. And that's actually what they did when they slaughtered the British cows, especially the ones that were infected with mad cow disease. They just incinerated them at very high temperature. Another thing they can do, and this is what they do in the hospital when they have surgical instruments, which they want to sterilize because they're worried that they may have been contaminated with prions, they will treat them with proteinases. And that does destroy prions. Okay. Because obviously even a scalpel, which is made of metal, you don't want to subject it to 1,000 degrees Fahrenheit, because I think the metal would melt at that temperature, but I don't know. Don't quote me on that. Anyways, it's too hot for scalpels. Okay. The last disinfectant are peroxygens. These are oxidizing agents. And this is a chemical disinfectant that does kill endospores. So there are two chemical disinfectants that do kill endospores. I'm talking about all endospores. And peroxygens are not that uncommon, and they're not that, I don't know, strong of disinfectants. Like hydrogen peroxide is a peroxygen. And we have that in most of our homes, and we use it on our skin. So you know it's not that toxic, at least to us, but yet it is very deadly to endospores. Part of the reason is endospores are produced by anaerobic bacteria, and peroxygens use oxygen or hydrogen peroxide or something like that that generates oxygen. And so they are very sensitive to peroxygens. Any question about any of that? And so if you wanted to kill endospores, an easy way of doing it that doesn't require much time is just to expose them to a peroxygen, such as hydrogen peroxide. Now, that's not to say that some of these chemical disinfectants can kill certain endospores. Like I know bleach, which is a halogen, can kill some endospores, but it does not kill all endospores. Some endospores resist the treatment with bleach. And so it does not kill at least some species of endospores. And I can't tell you what they are or why they are, but some of the endospores are much more resistant to killing with bleach than others.
One more question, Dr. Volpe.
Uh-huh.
So the autoclave also does not kill all the endospores, but some of the endospores, right?
Well, if you're talking about a 1 liter solution for an autoclave, the autoclave will kill the endospores in 1 liter. But if you have more than 1 liter, like let's say 10 liters, and you put it in the autoclave for 15 minutes at 121 degrees, I don't remember the pressure, something like 15 pounds per square inch, it would not kill all the endospores. So I'm answering it that way because the autoclave does start killing endospores. And if you have less than 1 liter, the endospores will be killed. Okay. And most of the time when we put liquids in the autoclave, we don't have more than 1 liter. And what you can do if you have more than 1 liter is you can just autoclave for longer than 15 minutes. Now, obviously 10 liters might require 10 times the time, and that would be 150 minutes. That's starting to become fairly long. All right, any questions about any of that? If not, here's a table summarizing the different chemical agents, the mechanism of action, the preferred use. You should only focus on these 3 parts of the table. I'm not going to go through this other than discuss the agents, phenol, phenolics, bipheноls, biguanides, halogens, alcohols. The preferred use, I'm not going to even test you on that, but with halogens, the preferred use for most of the halogens are as a disinfectant, like chlorine, bleach. The exception is iodine, which is a halogen. And, you know, we don't use that as a disinfectant. We use it as an antiseptic. We apply it to skin. And I don't know why they don't use that as a disinfectant, maybe because it's more expensive and bleach is similar and cheaper and it's more effective as a disinfectant. Alcohols, heavy metals and their compounds I do want to mention something here, and that is the use of heavy metals has come back into the hospital for a long time. I don't know when, but let's say 1970s to about 2010, we stopped using heavy metals because, well, they are toxic to humans, and we had better agents that we could use. But now, because bacteria have become resistant to antibiotics, we are starting to use heavy metals again because the bacteria are not resistant to the heavy metals. And they are effective at killing bacteria. Like I said, the main reason why we stopped using was simply because they were toxic to human and we had better agents. But now we use heavy metals all the time in like hospital dressing, or when somebody gets a wound infection and they constantly get the wound infection, so they don't want to give antibiotics because the patient will become resistant to those antibiotics. Surface active agents, these are skin degerming and removal of debris like hand soap, antiseptic for the skin, instruments, utensils, rubber goods. There we go, soaps and detergents, acid and anionic sanitizers, quaternium ammonium compounds, which are used frequently in the hospital. I guess an antiseptic for the skin. The chemical food preservatives, we can add organic acids, and I'm sure you're familiar with this. Nitrates and nitrites are added to food to prevent bacteria growing. Aldehyde like glutaraldehyde, formaldehyde. We disinfect medical equipment with these. Chemical sterilization such as ethylene oxide. We sterilize agents with ethylene oxide and other chemical sterilizers when the agent would be damaged by heat. So most of the time we sterilize with an autoclave or put things in the oven. But if it's damaged by heat, we will sterilize with chemical sterilization like ethylene oxide. Like tubular and medical instruments. And then fluids. Peroxygens, the main one I'm going to talk about is hydrogen peroxide. We use it on our wounds. It is very effective against oxygen sensitive anaerobes, including endospores. Now, when we use the agent, it can have different effectiveness, like soap and water, and nobody washes their hands for 2 minutes, you're only killing about 90% of the bacteria when you expose them to soap and water. So it's not very effective. Any question about any of that? Tincture zephyrin, which I don't even know what that is. Tincture of iodine, that's iodine solution. When we expose that, our skin to that, it's very effective at killing 90% of the bacteria in about 90 seconds. And usually when you apply tincture of iodine, like to the skin, it's covering the skin and then you leave it there until it dries. So it's on the skin for a long period of time. And that's why tincture of iodine, or just iodine, used to be very common before surgery in the hospital. There are other agents that they do tend to use now, but iodine is still used. And in the home, iodine is used quite frequently. So there are different effectiveness. Another one that's not terribly effective, but it's between soap and iodine would be alcohol. Okay. Alcohol does not kill all bacteria, even for long-term exposure. It doesn't kill any endospores, but it does remove many bacteria. And so we call it more a degerming agent rather than an antiseptic. Microbes have different characteristics. And when we're controlling them, we can organize the microbes by their characteristics for how effective they are to the treatment. And when we're talking about the most resistant microbes for treatment, the most resistant is a prion. Prions are very hard to inactivate. And then the least resistant would be viruses with lipid envelopes. They're very easy to inactivate. And actually, coronavirus is a virus with a lipid envelope. So simply washing your hands will inactivate that virus, because soap is very effective at removing the lipid envelope, and that will inactivate the virus. And of course, soap would not do anything to prions, but most things would not do anything to prions. Endospores would be the next most resistant microbe to all of the disinfective agents. Mycobacteria, the next most resistant. Mycobacteria have mycolic acid in their cell wall, and that prevents any agent that is water soluble from getting in the bacteria and then damaging the bacteria. The cystiprotozoa and vegetative protozoa are next most resistant. Gram-negative bacteria, the next most resistant, and they are more resistant than the gram-positive bacteria because their outer membrane prevents some agents from getting through the outer membrane, going in and damaging the cell. And we did mention this a little bit when we're talking about penicillin G. It cannot get through the gram-negative cell wall and damage the cells. But penicillin G can damage the gram-positive cells. What would be another example? I'm drawing a blank here on another example other than another antibiotic that would be stopped by the gram-negative cell wall. But there are other agents. Here we go. The host immune system has more trouble with the gram-negative bacteria than attacking the gram-positive bacteria. So the host immune system can more easily kill the gram-positive bacteria than the gram-negative bacteria. Fungi, including most fungal spores, are more resistant. Viruses without envelopes are more resistant than viruses with envelopes. Gram-positive bacteria. And then, like I stated, the least resistant, the most sensitive to any chemical agent are viruses with lipid envelopes. Now understand this is a simplified hierarchy of the relative resistance of major microbial groups to chemical biocides. Realize that specific agents or specific species will be sensitive to certain agents. For example, mycobacteria are resistant to most of the antimicrobial treatment, but mycobacteria are very sensitive to alcohol. because the alcohol goes right through the macolic acid. And then it damages the cell wall and damages the cells. And so mycobacteria are very sensitive to alcohol. And yet alcohol is not that good of an a disinfectant against microbes. So alcohol is a very weak disinfectant. That's why we call it a degermer instead of a disinfectant. But it is good at killing mycobacteria because of the characteristics of mycobacteria. The macolic acid makes the mycobacteria very sensitive to alcohol. Any question about that? So that's the one example I'm going to mention. I also mentioned about endospores. There's few of the biocides which are effective against endospores, but the endospores are very sensitive to peroxygens, which use oxygen to kill the cells. And because all of the endospores are made by anaerobes, the endospores as well as the vegetative cells are sensitive to peroxygens. Any question about any of that? We talked about viruses. Let me talk a little bit more about prions. They are the most resistant microbe. They're not really alive, all they are proteins. How do we totally inactivate prions? Well, it's not completely solved yet. But two ways they have come up with are treating, like the mad cow, with 1,000 degrees Fahrenheit, incinerate the carcass. And the other way would be to treat with proteinases, which it'd be impossible treating the cow with proteinases. Hopefully in the hospital, they don't have a prion infected patient, because if they were to do surgery on the prion infected patient, they really have to go to extremes to remove the prions from the surgical instrument, the operating room. Prions are difficult to kill. All right, so the final comments on the controlling microbial growth. While microbial control methods are effective, especially the biocides, they are not uniformly effective against all microbes. The compounds discussed in this lecture are not generally useful in disease treatment, with the exception of the antibiotics. Any question about any of that? So what biocide is the best biocide to use? And we're going to say best: cheap, very effective, kills most microbes, easily obtained, easily used. What biocide is the best? Nobody's got a guess? Well, bleach is one of the best because it's easy to obtain, it's cheap, it's very effective to use, but it does have some drawbacks, and that is it is inactivated by organic matter. And the other drawback is that it's not effective on all endospores. It's only effective on some endospores. To kill endospores, you want to probably use a peroxygen, such as hydrogen peroxide. And so the best biocide will depend on what agent you're using the biocide on. Any question about any of that? All right, If there's no questions, let's begin the review for the final. I think I'm going to start talking about the reverse. So I'm going to work in reverse. Chapter 7, well, essentially you should know everything in this chapter because you haven't been quizzed on it, but I'll try and point out some things to focus on. And what would be the best way to do this? Let's go to the study guide. I apologize, this is not going to be very large in print. Well, you should always know the terms for each of the chapter, especially for the last chapters that you've not been quizzed on. You should know all the terms because I could ask you for the different terms. You should be able to discuss the factors that influence the rate of microbial death. You should be able to discuss the three main ways in which microbial agents kill or inhibit microbes. What are the three main ways that microbial agents kill microbes? Anyone guess?
By puncturing the plasma membrane.
Yeah, by puncturing membranes or damaging the membranes, that's one. What's another?
Is it like when the white cells called like phagocytosis or something like that? They like envelop it.
That would be how the host defends itself against microbes, but we're not talking about that. We're talking about three ways that a microbial agent kills or inhibits the microbe. All right, the three ways are damage the membranes, damage the protein, meaning inactivate or oh, what do you call that? I'm losing it here.
Denaturing.
You inactivate the protein, you coagulate it, or you
Denature.
denature, thank you. Denature the proteins. So that's the second way. The third way is to damage the DNA or the nucleic acid of the genome for a virus. So damage the genome. Okay, those are the three main ways that microbial agents kill or inhibit the growth of microbes. Be able to describe the application of the following physical methods for microbial control. Tell me about heat. There's two types of heat, moist heat and dry heat. Be able to tell me which one is more effective. Moist heat involves autoclaving and pasteurization. Dry heat, there's a dry heat sterilization, such as baking it. And then there's very high temperature, such as incinerating it. There's also filtration, low temperature, which generally does not kill microbes. The low temperature just stops their growth. High pressure, desiccation. This does kill many cells, but not all cells. For example, a cell that has an endospore will be fine with desiccation, but something like E. coli falling into molasses, it will die. Osmotic pressure and then radiation, and you should be able to discuss ionizing radiation, non-ionizing radiation, and you don't need to know about microwaves, okay? So be able to discuss the pros and cons of each of these methods for controlling microbes.
I have a question for the radiation.
Uh-huh.
So the ionizing is with the shorter wavelengths or is it with the longer wavelengths?
I'll have to look that one up. I don't remember the wavelength of UV compared to the wavelength of ionizing radiation. If I remember correctly, and don't quote me on this, I think ionizing radiation is shorter than UV light, but let me look that up to be certain on that. And you don't need to know the wavelength of the radiation for the exam. Where is that? Oh, that's chapter 7. I already got that. That's right. Here we go. So, let me blow this up. This is the wavelength. And so the shorter wavelength is ionizing radiation, and UV light is a longer wavelength. So I got it right. Be able to describe the factors related to effective disinfection and diffusion method for evaluating a disinfectant. And remember, we did that sort of in a lab where we had the diffusion method and then rated how effective, well, we rated specifically how effective the antibiotics were, but you could apply the same technique to rating the disinfectant. Okay, as far as the types of disinfectants, you only need to know the types of disinfectants that we discussed. So that would be chlorine, bleach, tincture of iodine, alcohol. Was there any other I discussed?
Phenols,
I didn't really discuss phenol. I mentioned the name, but I didn't discuss how it worked or how effective it was. Understand the relative resistance of the major microbial groups to chemical biocides and be able to explain why gram-negative bacteria are generally more resistant than gram-positive bacteria, and why envelope viruses are less resistant than non-envelope viruses, and then why endospores and prions are very resistant to chemical biocides. Any question about chapter 7? All right, let's move on to chapter 13, I think was the last. Well, that didn't seem to work. Let me open in a new page. And then I'll have this one. Viruses, viroids, and prions. You should study this lesson very well because we haven't been tested on it. Be able to describe the differences between eukaryotes, prokaryotes, and viruses. Be able to discuss what general characteristics all viruses share. And that would be they all have a genome. They all have a protein coat. Be able to describe the composition and the structure of an envelope and a non-envelope virus. Be able to describe the composition and structure of a bacteriophage. And well, we got it down here. be able to describe the bacteriophage lytic life cycle, and be able to describe the bacteriophage lysogenic life cycle, and be able to discuss the major difference between the lytic life cycle and the lysogenic life cycle. Anyone remember what the major difference is?
The lysogenic is in symbiosis,
Symbiosis,
and
the host cell,
yeah, and then
and the lytic life cycle virus. Are we going to mention it? The lytic life
it-
cycle virus is not in symbiosis. It lyses its host cell.
Yeah.
Okay. Be able to answer what is a prophage. For a bacteriophage, we only have prophages for the lysogenic life cycle. Is that true? Yes. With animal viruses, we can have a prophage outside of a retrovirus or something, meaning you can have a prophage happening in other life cycles of an animal virus. Now a lytic virus, can there be a prophage in a lytic virus? I don't remember. I can't think of any off the top of my head, but all right. Be able to describe the structure and the multiplication cycle of the following: a DNA virus, an RNA virus, and then there's two types of RNA viruses: a positive strand RNA virus and a negative strand RNA virus. We're talking about single strand RNA viruses here. Be able to describe the structure and the multiplication cycle of a retrovirus, and be able to answer what is a provirus. Provirus is, of course, when the viral genome integrates into the DNA of the host cell. And when a provirus forms, the provirus will remain in the chromosome of that cell for the lifetime of the cell. Understand how a virus can lead to the formation of cancer. And there are two ways a virus can do that. One is with an oncogene. So an oncogenic virus carries an oncogene to the cell, and then that oncogene changes the cell so that the cell replicates uncontrollably, and that can lead to the start of cancer, meaning it may not explain totally the cancer, but at least starts the cancer going. Are you understanding what I mean there? I don't really want to talk about cancer here, but cancer is caused by not one mutation, but something like 5 or 6 mutations. And the oncogene starts the process. Okay, so that's just one of the 5 or 6 changes that the cell needs to become cancerous. Any question about any of that? The second, go on.
I have a question regarding the positive strand RNA and the negative strand RNA. So the negative strand the ribosome cannot read the negative strand, right?
Correct.
And then from the positive strand, it can directly transcribe as an mRNA.
Correct.
It's a little confusing. Just wanted to clarify.
Okay.
Thank you.
All right. The second way a virus can lead to the formation of cancer is when the virus integrates into the host cell's DNA. It can muck up the DNA. And for simple reasons, we'll just say it can mutate the DNA. Okay. Be able to describe a latent and a persistent viral infection. Be able to answer what are prions and what do they do meaning how does the prion protein replicate in a cell? And you should mention what are some prion diseases, like mad cow disease in cattle, also called bovine spongiform encephalopathy. And the main one in human is Creutzfeldt-Jakob disease, but there are a few others in human. The only one I recall off the top of my head is Kuru. And it'd be able to explain how they are transmitted. Most of the prions are transmitted by consumption of the prion, and not usually consumption of the prion itself, but prion tainted meat is most likely the transmission of prion disease. You can get it in other ways, like you can get prion disease from an organ transplant or from a surgical instrument or from a needle puncture. Or why you should not share needles. Of course, you can get other diseases besides a prion disease. But if you were to share a needle with someone, you could get prion disease. And then briefly describe a plant viruses and a viroids. And note that viroids are only found in plant cells. All right, any question about any of that? If not, let's move on to chapter 14. Make sure that's turned on. Looks good. Oh, that's not what I wanted. So in chapter 14, please learn the terms of this chapter. If you learn the terms, you will learn about half of the chapter, because this chapter is very, I don't know, dependent and very strong on terms. Be able to describe the normal microbiota and how are the normal microbiota beneficial to us. We should be able to know that. I could ask you, how can the normal microbiota benefit us? How does the normal microbiota help us humans?
Protect us.
It can protect us by preventing the establishment and growth of a pathogenic microbe. And it can use up the resources so the pathogen can't get established. It can simply occupy the space so the pathogen cannot get established. It can produce a bacteriocin, which would kill the pathogen. It can change the pH or the environment, making it unsuitable for the pathogen. And that actually happens in our mouth and on our skin and our urethra, where the normal microbiota changes the environment, making it less suitable for a human pathogen to grow. Let's see, of course, the acid in your mouth isn't good because it weakens your teeth or the enamel on your teeth. Oh, there's one other way that they're beneficial, and that is they can help us by providing vitamins and not really minerals, but vitamins and help us digest our food. So the microbes in our gut really help us digest our food. Know what is meant by the term symbiosis and the three types of symbiosis: commensalism, mutualism, and parasitism. Know that the normal microbiota are usually either commensalistic or mutualistic. And then a human pathogen follows the parasitism symbiosis, meaning it harms us and it benefits from living off of us. Be able to describe the relationship between the normal microbiota and an opportunistic pathogen. Normally, the microbiota do not cause disease in healthy individuals, but they can under certain circumstances, and then we call it an opportunistic pathogen. And that would be like Staphylococcus epidermidis not growing on the skin of a patient, but getting in a puncture wound or something, and then growing in the puncture wound. Realize that people who have a defective immune system, such as a AIDS patient, can have the normal microbiota cause an opportunistic infection. Whenever the normal microbiota is growing where it should not grow, it would be an opportunistic pathogen. And that could be like E. coli growing in the ear, or E. coli growing in the bladder. E. coli shouldn't be in those locations. Be able to discuss Koch's postulates. We discussed this 3 times this term. So you know there's going to be a question on Koch's postulates on the final. How are Koch's postulates used? And why are there exceptions to Koch's postulates? Koch's postulates are used to associate a given organism with a given disease. But sometimes there's exceptions to using Koch's postulates, and that would be for an organism, for example, that we cannot get to grow in vitro, like syphilis or Bartonella henselae, which causes cat scratch fever. We can't grow it in vitro, so we can't use Koch's postulate to show that this bacteria causes these diseases, or this disease. And then whenever it's unethical to use Koch's postulates, like we've never injected HIV into humans to determine that HIV causes AIDS. Be able to describe the ways of classifying infectious diseases, be able to explain the terms related to the occurrence of disease, understand how the severity and duration of a disease is described. So these are all terms. Be able to explain why vaccination and herd immunity are important. Be able to explain the extent of the host involvement in infection. Be able to explain the usual patterns and predisposing factors of disease. Be able to discuss the different stages of the development of disease. You should be able to discuss the incubation period, what's happening, the prodromal period, what's happening. The incubation period is the time between when a patient is first infected with the pathogen. Between that time and the time they first get symptoms. Prodromal period is when the symptoms first appear and they're mild till the time when the symptoms and signs are severe. The period of illness begins when the signs and symptoms become severe. Generally, the patient is at their sickest during the period of illness. Generally, this is when the pathogen is at its highest numbers in the patient. The period of decline is when the patient starts getting better. The period of convalescence is when the patient has gotten better to the point where the infective agents have decreased to the point where it reaches a threshold. Let me try and draw that. Close that. I probably should have closed that too. I'll leave that one open. So when we're looking at the number of organisms on the y-axis and the time on the x-axis, generally speaking, the microbes will increase from the first period and will be at their highest during the period of illness and then decrease. And there's this threshold here where below this threshold, the organism is at low enough numbers that you don't have any signs or symptoms. And so we see this in the incubation period and then the period of convalescence, where you still have the organisms. So this period right here will be in the period of convalescence, but the number of organisms in the patient are below the threshold, so the patient has no signs or symptoms that they carry the microbes. Any question about any of that? I meant to close that. Be able to describe how infections can spread via human, animal, and non-living reservoirs to a susceptible patient or susceptible host. Be able to discuss the various modes of transmission that spread disease. Understand what nosocomial infection is. And let me state that you should know this. I will definitely quiz you on nosocomial infection because this is one of the most important things you can learn from microbiology. A nosocomial infection, also called a hospital-acquired infection, is one the patient gets simply from staying in a hospital-like setting. So a hospital or hospital-like setting, like a nursing home or something like that. And if they had not come to that hospital-like setting, they would not get the nosocomial infection. I'll be able to list the 3 factors involved in a patient getting a nosocomial infection. First, there's microbes in the hospital-like setting. Second, the patient is immunocompromised. That's why they're going to the hospital-like setting. And then third, there's a chain of transmission of the nosocomial infective agent in the hospital-like setting. And then learn how nosocomial infections can be prevented. I will mention that 50% of nosocomial infections can be stopped simply by frequent and thorough hand washing. Be able to discuss what an epidemiologist does and be able to define morbidity and mortality. Any questions about chapter 14? If not, let's move on to the earlier chapter. Let's briefly talk about lab 9 because you will be quizzed on lab 9 for sure. You'll be quizzed on the other labs, but lab 9 more so than the other labs. You should know what IMVIC is and know that the test, indole test, the methyl red test, and the Voges-Proskauer test, and then the Simmons citrate test is what IMVIC stands for. You should know when the test is positive for indole, methyl red, and Voges-Proskauer, the test reagent will always turn red. And then for indole, methyl red, and Voges-Proskauer, it'll be a yellowish color if it is not positive, meaning it's negative. Any question about any of that? For the Simmons citrate test, if the tube turns blue, that means it is alkaline. Then it is positive for the Simmons citrate test. You should know about carbohydrate fermentation. We have a tube, which we call the PR carbohydrate test, and it'll have one sugar in the tube, which can be fermented. And if the sugar is fermented, the tube will turn yellow. It turns yellow when acids are produced, and acids are produced when the sugar is fermented. You can also have gas production if fermentation occurs, in which case if it's fermented in positive, we say it's A. And if it is fermented in positive and gas is produced, we say AG, AG standing for acid and gas. So that's the carbohydrate fermentation tube. You should know motility, and motility can be studied in more than one tube. In the motility medium, you stab inoculate the tube, and if the growth is only in the stab mark, so you have to do a good stab, so the growth is only in that one stab mark. If growth is only in that stab mark, it's because the bacteria is not motile. If the growth flares from the stab mark, or if the growth is everywhere in the tube, which means it is looking opaque and cloudy everywhere, then the bacteria is motile. And if it's only flared, it means the bacteria is slightly motile. And if it's everywhere and cloudy, it means the bacteria is very motile. Motility in bacteria, recall, only happens if the bacteria has flagella and then can swim through the media. In motility medium, we do have agar in the media, but the agar is at very low concentration, enough to make the agar like jello, but it is semi-fluid and the bacteria can swim through it. There's the carbohydrate fermentation tubes. I guess I'll mention that because we just talked about it. And it includes an inverted Durham tube, which has media inside of it. And if gas is produced from fermentation, you'll see a bubble in the inverted Durham tube. We can assay motility from the motility medium, but we can also assay it from the Sims media, shown here. And shown here. Once again, you see the bacteria everywhere in the tube. So this one, sorry, I can't blow this up too well. That's a little better. We see bacteria growing almost everywhere in the tube. Certainly it's on the right side. So this is a motile bacteria. If the bacteria is only growing in the stab mark of the Sims tube, then it's non-motile. Any question about any of that? Oh. Oops, where am I? There we go. You should know about tryptic sugar agar, and this test can determine if the bacteria produce hydrogen sulfide. If it turns hydrogen sulfide, the tube will turn black at the bottom. You should stab inoculate the TSI or triple sugar iron, as well as grow the bacteria or inoculate the slant. And then we're looking at growth aerobically on the slant, and then growth anaerobically in the stab. For assaying hydrogen peroxide, you can assay it with TSI, as I mentioned. You can also assay it in the SIMS tube, that's sulfur indole motility tube. Once again, if hydrogen sulfide is produced, the tube on the bottom will turn black. So that's the same in tryptic sugar iron or TSI, and then SIMS, it turns black. The third way to assay for hydrogen sulfide production is to grow it on the what is it called? The HE media. And if the bacteria form black colonies, it's black because hydrogen sulfide is being produced. Any question about that? All right, I talked about TSI. You should know about TSI, and let's talk about it here. No change. The color will remain red, or it'll change pink on the slant. It becomes pink because it's more alkaline. If it is all yellow, then in the slant and in the butt, then the sugar glucose is fermented, as well as one of the other sugars, either sucrose or lactose. If it is only yellow in the butt, but it's pink in the slant, then only the sugar is fermented, only the sugar not fructose, glucose is fermented. Glucose is at lower concentration, and when it grows aerobically, it used up all the glucose. So if we had looked at this earlier, the slant would have been yellow. But once the glucose gets used up, it starts digesting, the bacteria starts digesting protein, which produces ammonia, which is basic, and that will change the color of the slant to pinkish. And so this is glucose being fermented, and only the sugar glucose. And you can tell that because the butt is yellow and the glucose is fermented slower anaerobically than aerobically. Let me mention one more time that if the glucose is fermented and gas is produced, you'll see cracking of the media as well as a bubble. You might see a bubble if there's enough gas produced. And then lastly, on the tryptic sugar agar, if hydrogen sulfide is produced, it'll be black on the bottom. In this case, this is yellow, you just can't see it because of the black. And generally, if ever it's black, the sugar is fermented. In this case, only glucose is fermented because the slant is red, the butt is yellow. All right, any question about any of that? All right, you should know that HE only grows gram negative, so if it doesn't grow, it's a gram positive. If it grows, it's a gram negative. The same with McConkey's. If it grows, it's a gram negative. On McConkey's, if it turns pink, it ferments the sugar lactose. On HE, if it becomes salmon colored, it ferments the sugar lactose. And then it's non-colored if it does not ferment the sugar lactose. All right, for lab 9, we did some case studies, so you should be able to answer the case studies. If I give you a case study and a multiple choice question, it'll be fairly simple so that you can finish the question in a minute or two. Okay, if it's an in-depth case study, I'll only have it in the essay type questions. Because I expect that it'll take you longer than a minute or two to follow, finish it. Okay. Any question about any of that? All right. We talk about chapter. I can close that. Did we talk about chapter 13? Yes, we did. Chapter 14. All right. I will continue talking about the chapters that we have already been quizzed on in the lab at 6:30. Any questions?
Yes, so would we go over that on Tuesday as well? For the
No, there is no class on Tuesday. That's finals week.
Okay, thank you.
I do have a quick question about that. In Canvas, it shows that there's a Zoom meeting with Anna Tori on Tuesday. Should we disregard that?
I'm not familiar with this. I'm not sure why there would be a Zoom meeting with Anna Tori. Where is that?
It shows on the dashboard, at least in my Canvas. And
Yes,
it's like a Zoom meeting link.
Let me take a look at that. I don't know anything about that.
Okay.
I don't know why Anna Tori would have a Zoom meeting.
Okay.
Okay. Where do you see that?
Just on the dashboard.
Yeah, I'm on the dashboard.
Yeah, it just shows up for me on Tuesday, the 14th. I'm not sure if anybody else is seeing that on theirs.
Is it on the right?
It's basically like in the to-do list.
To-do list. I
And
don't
it
have
could,
a to-do list, so I can't
Okay.
verify it.
well, I just wanted to make sure. So that's perfect. I just won't worry
I
about that.
don't think there will be a meeting. Anna Tori. I mean, I'll check with him, okay? But
Okay.
I thought it was last term, Anna Tori finished my class. And so this might be something that he did last term,
Gotcha.
okay? But I don't know why he would have a Zoom meeting, so I'll check with him.
Okay.
I don't see it on my dashboard.
Yeah, it shows up on mine too, under that to-do place, but then also in the calendar.
Huh? Okay,
yes, on mine too.
let me see if it's here. I got 5 more things. Nope, I don't have anything there. Let's take a look here. Oh, here, new lecture Zoom, Anna Tori. And then there's a lab one I've got too, June 14th. Let's see what happens when I click on that. Nothing. It just brings up the calendar. All right, like I said, I don't think we'll have a Zoom meeting with Anna Tori, but I will check with him and get back to you. So I'll make it as an announcement. And I think I made a new announcement. I guess I should mention that. It's not terribly important, but there was an announcement about they wanted microbiology tutors for the tutoring center. And so I made this link here. You can get paid if you're willing to become a microbiology tutor. And that would be for next term or some future term. Okay, any questions? If not, I'll see you in the lab.